Selectis is a gene editing company. From the start in 1999, Selectis was founded with the idea of modifying gene, meaning even at that time, the term gene editing didn't exist, but the main goal of Selectis was really to be able to modify gene and cure diseases. So that means all the projects that we are doing at Selectis takes their roots with the gene editing technology. So everything starts with a gene editing tool. So actually we are pretty agnostic in terms of technology. And in the past 21 years, we have been tested many of the technology that has been developed. This is including TALEN, CRISPR, meganuclease, megaTAL. And at the end, we have selected the TALEN as the most powerful technology. Why? Just because it's the most precise, efficient, and accurate technology. And we think that it is the most adapted for the therapeutic use.
When we have a sequence of interest on which we want to design talents, we scan the sequence of this sequence with a machine learning model that has been trained over the year with all the data we have accumulated, and it will determine the sequences that have the highest probability of having a good activity. This machine learning model uses both sequence features as well as epigenomic features to determine the highest probability sites. This process takes place in a matter of a few dozens of seconds. In a second step, we will look at the specificity of the design talents. For that, we will use a second model that will scan the genomic sequence, look at the homologous sequences, and evaluate the probability that these sites are cut. Once we have determined the best candidates, we will send them to the New York lab so that they can synthesize them.
Hello, welcome to Selectus, New York. Today we will show you in this facility how we go from lead talent candidate to next generation UCAR-T prototypes. Please follow me. Once we receive the designs for the TALEN that we need to assemble from bioinformatics, we then use the robot to assemble the TALEN from the starting material. We have the raw materials that allow us to assemble essentially any TALEN we want against any target we want. The robot that we use is amazing as far as I'm concerned. We do many different things with it, and this robot gives us a lot of flexibility and a lot of power to do things efficiently, quickly, and in larger scales than we were able to do previously. We assemble the TALEN, we produce them. Here in New York, we use next generation sequencing, which we have here in-house, that we use to test the TALEN from start to finish, from activity through specificity to, in the end, pick the best one. And then from that, we generate a large quantity of RNA to then pass on to the next person for future tests in whatever experiments they're doing.
So the innovation team spends most of their time in the BL2 lab, that means Biosafety Level 2 lab, where is the place that we are allowed to work with viruses, and is the place where we can modify these blood cells from the patients to express the CAR, which is delivered via a virus. So after we have incorporated the virus into the blood of the healthy donors, and specifically into the T cells, we use an electroporator to incorporate the TALEN into the cells. And once we bring it in, the TALEN will express and will be able to modify the genome and generate the modifications that we want, the modifications that will make also each of our UCAR-T cell products unique. And then once we validate that we have all the edits and all the modifications that we need, then we need to expand the cells, because that's the beauty of the allogeneic CAR-T cells, to start with a small number of cells and to end up with a large number of cells, so to generate multiple doses. And then when we know that from a small number of cells, after we modify them, we are able to get a large number of cells, is when we have our prototype and we are ready to send it to the next level.
Welcome back to Paris. We will now show you how our process and analytical development teams work hand in hand to prepare scalable production processes. And as a sneak peek, we will also show you our GMP facilities where Selectis is now producing critical raw materials and the gene editing tools we need for our manufacturing.
Along the life cycle of the product, the development work comes in between the innovation and the final clinical manufacturing. Our main mission, together with the analytical development group, is so to start from the proof of concept that has been made in New York, delivering us the talent and the core construct, to put in place a robust manufacturing process to allow the production of UCAR T cells. So really the main mission of our group is to upscale robust methods, efficient methods that are also suitable for clinical manufacturing, so easy to implement in a GMP environment. That process, years ago, was designed for a very first product, which was UCAR T19, and we decided to slice it down into different unit operations that can be assembled together, put one after another, and then we assemble them, of course, into a global process, what we call today some kind of a platform. Every time we try to use the same global backbone and the same unit operations. However, we also need to fine-tune them depending on the product. And that fine-tuning is very dependent on what the analytical method are telling us. At every step, we look at the cells, we test their activities, and so we are working really closely with the analytical development group because they give us all the methods that allow us, again, to make sure that what we do fit what we want to do.
In parallel to the development of the process, we are developing all the assays that will be able to monitor and characterize the quality of the UCAR T batches. Among others, we are developing identity, safety, potency, and purity assays, and these assays will be developed and pre-qualified according to the guidelines from the regulatory agencies. All these assays are performed using flow cytometry, and one of the tasks of my team is to put in place the antibodies panels that will allow this characterization. Eventually, this assay will be transferred and implemented in the QC lab at Raleigh in a more controlled and qualified environment. This assay will be finally qualified and validated, and then will be used in routine for the release of the GMP batches for clinical use.
The GMP Paris production, which is, I will talk about this more, it's the production of our raw materials, our starting materials. This activity is a GMP activity because we use those materials in cells which will then enter clinical trials. So therefore, it's important that the starting materials are made according to good manufacturing practices. This means we make them in biobenches, in isolators, and of course in clean rooms. As you can imagine, we started manufacturing some of those steps, the cell banks, the plasmids. Now the next step will be the mRNAs, and the next step, which we are preparing in parallel, are the vectors. So now those products will be transferred by cold chain to Raleigh. So this is very important that the temperature are respected and that during the transportation, the low temperature needed for the conservation of these raw materials is respected, and that we can document that this low temperature has been respected all the way from Paris to Raleigh.
Hello, welcome to Selectus Raleigh. In here, you will discover the new wave of gene editing and how we apply it to manufacturing. Come on in.
This Raleigh facility is Selectis' new state-of-the-art biomanufacturing plant. It's designed to take the processes that were created by our innovation scientists in New York and in Paris and optimized and scaled up by our development team in Paris and duplicate those here at GMP manufacturing scale to make product that can go to patients. The materials are received from Paris and entered into our inventory system, inspected, and stored in the GMP warehouse or controlled storage temperature chambers. They require low storage condition for the talons and vectors, so we have a number of different types of chambers at different temperatures that are appropriate for different types of materials. So once the materials have been inspected, inventoried, and put in the warehouse, they are taken out of the warehouse and staged into manufacturing as necessary during the 3-week production process. So different materials come from the warehouse into manufacturing as needed during this 3-week process. I and the team here are very motivated to make this biomanufacturing facility a reality and a big success for Selectis. We're going to have control of our own destiny by having this manufacturing facility and being able to maintain supply to patients that need life-saving therapies.
So construction has been a big challenge, but one that we've really successfully met. We started with a shell of a building, have renovated it almost completely to incorporate all of the clean rooms and isolator technology that we need to manufacture Selectis' products in a very unique and innovative way. So Selectis has used industry standard equipment and adapted it for our process, as well as designing equipment specifically for our cell therapy process off the shelf. And that equipment is made for cell expansion, cell washing, cell selection, and then some of the custom designed equipment we use for doing the electroporation process to do our cell engineering. So the security of our cells is critical. Throughout the process, we make sure that we have chain of identity managed, as well as manufacturing inside the isolators to ensure that we have produced the most safe and pure product for our patients.
To build a true GMP manufacturing facility, you have to be exacting and specific about everything. There should be no activity that you do in a GMP space that doesn't have a process underlying it. And in order to support that, you have to build a facility that allows for all of these things to happen in an efficient fashion. So we receive an incoming collection of frozen cells. We thaw those cells. We use a lentiviral vector to transduce those cells. Then we electroporate those cells along with some talon to remove a specific section of DNA. From there, the rest of the process gets really simple. We expand the cells, we grow as many of those cells to make as much treatment as possible. We purify those cells to select for just the appropriately gene-modified T cells that we want to infuse into a patient. Then we move to the robot. Here we have an automated system that fills a series of vials, laser seals the filling holes, caps those vials. We inspect them, we freeze it down, and then we store it long-term until the first patient needs their treatment.
At this site, we don't just manufacture, we do manufacturing and testing. During the process, but most importantly, at the end of manufacturing, in order to know that the batch is safe and efficacious, we have to do a battery of tests for release. The certificate of analysis lists all the tests that are required, what criteria they have to meet, and what results did you get. So after QC does the testing and assembles all of that information in the certificate of analysis, QA will review that and determine that the batch is acceptable or not, and they will release the batch for use. So it can't go out the door unless it has been tested and passed all of its quality control tests. If we don't have the QC tests that show that we meet the requirements of the certificate of analysis, we don't have a product. These products are the most challenging that I've ever worked on. The assays are the most complex. The products are the most complex because they're living cells, so it's not just a single molecule or some inert thing that can be highly variable and complicated to assess. I've always worked in science, and most of the time I've either worked in rare diseases or in serious illnesses where it truly impacts their life with the products that we make. And I found that never to be more so true than when working with gene therapy, cell therapies, and tissue therapies. because these products are life-saving products.
We've been really excited to have toured you this week through our Selectus innovation cycle. We are innovators. We break paradigms. Our goal is to revolutionize molecular medicine in the 21st century. This is our commitment to a cure.
Nobody will explain you what the next wave of Cellectis gene editing technologies is. Come and see for yourself. Join us tomorrow.
Hi and welcome everyone. Welcome back to those who have already followed the first few episodes of the Selectus Innovation Days. I'm Simon Harness, the Chief Investment Officer at Selectus, and I'll be moderating again today's Q&A session. Again, as a reminder, you can just type in your questions in your live webcast and we will see them here, and I will summarize them and direct them to our different teams. We have on the line today from Raleigh in North Carolina, from Paris, France, and we have a few of my colleagues here in New York City today. So we're super excited to share this manufacturing insight, really for the first time that the world is seeing how we are actually manufacturing our CAR T cells and what's under the surface in Selectus. The first question here from Jack Allen at RW Baird is about cost of goods. Can you talk a bit about the cost of goods for allogeneic CAR T? What do you anticipate the cost per dose will be for UCART product, and how will this compare to autologous products? And this is a good question for Steve and Raleigh. Please go ahead.
Sure, thank you for that question. Clearly, we can't get very specific about estimated and future cost of goods, but in the framework of allogeneic versus autologous, I will point out kind of the obvious to everyone that the cost of manufacture of a batch of allogeneic versus an autologous therapy is approximately the same. It's the same order of magnitude. The difference is that we are dividing that cost of a batch over hundreds of vials produced per doses, so that's going to have a very favorable impact on COGS. But in the future, we will be discussing more specifics about COGS.
Thanks, Steve. I have a follow-up question here from Ingrid Gaffenhau from Kempen. Over time, how do you envisage the Paris and Raleigh facilities working together? Would they work independently to cover different markets, or will each of them take a specific step in the process? And I will start maybe with Paris, and feel free to hand it over to Raleigh.
Well, Simon, I'm going to start with this. Hi, everyone. Hi, Ingrid. It's Andre speaking. New York, Paris, and Raleigh, as shown in the movie, works hand in hand. Currently, you have the speakers in Raleigh that are mixed between Paris team and Raleigh team all together working hand in hand, and that's what makes the power of having internalized everything in the company. And this power comes into a very powerful execution and an excellence in the production and the way we're doing the things. So yes, the three entities, with the travel restrictions, etc., we've been able to travel. We've been able to communicate, we've been able to exchange on a daily basis and an hourly basis, and that's what makes the thing. So tech transfer internally works with people traveling from one place to another, and that's what makes the things. We have our process development and analytical development team always in close contact with the production team So the osmosis works very well, and we've never seen this in the past. Transformation is real now.
Thanks, Andre. So there's a lot of questions coming in, and what I'll do is I'll summarize the questions that are of similar nature, and we will try to get through each and every one of those one by one. So maybe let's start with, again, a follow-up question from Kempen. Can you talk a little bit more about the process that happens before the CAR-T manufacturing? Where do we obtain our donor material from, and what are we looking at in terms of donor selection? And again, feel free, maybe Paris, you can start with that.
Okay, thanks for this good question. So it's here, Paris, Leo from Paris. So basically,
present yourself, actually, Leo.
Yeah, Leo Berthier, I'm SVP of Tech Ops here in Paris. Hi. So before making the CAR-Ts in Raleigh, there is the whole process of getting the starting materials. So the cells are come from a provider in the US, and then we have the production, the internal production of our plasmids, of the RNAs, and of the vectors. So for this, we have a capability in Paris, and this is very strategic for us because with this, we can decide how we plan the slots, the production slots. We also control the cost of goods, and we are much faster than, let's say, waiting for external slots with external providers.
Thank you so much. That's a great answer. And pivoting into more of the process, I'm getting a few questions in terms of how many products can we produce simultaneously, and also what is the capacity for the company and the timeline per product to manufacture from R&D to GMP batch. And maybe Steve and Raleigh, do you want to take that question?
I'm sorry, could you repeat?
Sorry, the question is the capacity of our manufacturing. So there's a few questions that are kind of around the capacity of our manufacturing. How many products could we theoretically do simultaneously? Does the capacity cover our clinical programs currently? And what is a rough timeline from concept to GMP manufacturing?
Sure. I'll let Dan Bright, our Director of Manufacturing in Raleigh, field that one. Thank you for the question.
Sure. So thanks, Steve. You mentioned Dan Bright, Director of Manufacturing here in Raleigh. Currently, what we're looking at is a minimum of 40 products a year per manufacturing suite. That's based on our current paradigm of manufacturing. As we expand and add additional suites, we'll be able to, for every additional suite, add another minimum of 40 lots a year. As we expand shift structures, we could potentially expand significantly on top of that. So quite substantial capacity based on the current way of working. And then to answer the second part of your question, we can process up to 4 products simultaneously in each individual room.
And just to follow up on this, what is the rough output at optimal efficiency that we think we can produce per product per year?
Well, that's a matter of campaigning and how many batches need to be produced. So the manufacturing capacity that Dan just described, we can allocate that according to what the product is and what is the patient demand, what's the patient population. So rarer diseases will require fewer batches. The rest of the capacity can be used for products that require more batches per year. But that ballpark of up to 40 per year per suite is our baseline operating at startup.
And just can you just precise, like, how many doses are we getting out of batches when you say 40 batches per year?
So the dose size is based on body weight. Minimum is dozens and dozens of treatments, right? Maximum goes up significantly higher, depending on the dose form, of course, as well. But you could look as high as over 100 doses per lot.
Great. Thank you so much. And here I have a follow-up question on more the technical aspect of this. What is proprietary about the electroporation device versus an off-the-shelf electroporator? this is a question for our, in-house technology that we have developed. I'll give this question to Paris.
Thank you, Simon. Thank you for the question. It's a good question because I think that this is a very differentiated competitive advantage that Selectus has, electroporation. In 2010, September 2010, Selectus purchased the asset of Cytopulse in Maryland, which was an electroporation company with a series of different devices. And since 2010, it's been 11 years we've been building up and improving these devices to fit our needs. What the difference between an off-the-shelf electroporation device and having everything internalized will be very intimately described tomorrow at episode 4 by David Sourdivin, the co-founder of the company that has been working on this with a series of different devices that we've been building over these past 11 years. And he will describe exactly what you can do. You can change the softwares, you can change the wavelengths, you can change the size of the chambers, you can change everything in order to fit your needs in terms of yields, survival, amount of nucleic acid or proteins that you want to put in there, the buffers that you would like to use, the conductivity. If you're using an electroporation device that is off the shelf, you're locked. You just have to push on the button and it's a black box in there. And this is not easy because every tweak, every need you have to go back to the company and ask the company to make the changes. You have to pay for this. It's complicated, it takes time, and there's a lot of requests over there because there is not that many electroporation devices. And here, this electroporation device is totally proprietary of Selectus, plus also, of course, our partners are using it with a lot of success, and the performance are totally outstanding and 100% fit to our needs. And that's what makes the difference. Selectus is independent and can do whatever they want. We want with our electroporation system and technology. So very good question. Thanks.
Thanks, Andre. Just in terms of these in-house technologies, are we looking into developing other delivery methods in-house, or what is the difference between the car delivery and electroporation?
Actually, we're working on a series of different technologies for delivery, and we're not down to this. The car is currently delivered through like standard procedure, which is the recombinant lentiviral vectors, which is standard like Kymriah or Yescarta or most of the CAR-Ts. This is a very big advantage because you can change the multiplicity of integration of different types, like the number of constructs that you would like to tune up in there. Plus, you're not totally dependent upon the expression of a single locus. For example, if you would like to knock in your locus at the TCR alpha, which seems a great idea, but TCR alpha is not expressed in gamma delta cells, so you lose gamma delta cells expressing the CARs. There are a series of different things that could be done this way. And that's why also lentiviral vectors give you a very vast array of integration and seems to be an extremely robust so far with very powerful ways to do this. So that's the way we're doing this. And we're working on more technologies to try to vectorize, and we're not going to go into the details in what you will have in the future. We need some meat for the next R&D days in the future.
And a follow-up question from Dominique at Merlin Nexus. Thank you, Dominique. What is the maximum number of edits or knockouts and CAR constructs that can be accommodated by the current manufacturing process?
I'm going to take this question again. So, hi, Dominique. Thank you very much for this question. It's appreciated. Dominique, we can do up to, we've disclosed yesterday in the session of the CAR-T, new preclinical CAR-T products, Mach 1, where there are 5 knockouts in the same product, not done simultaneously, but this is, and there is like 2 knock-ins in there, which is, I think, a spectacular result. And the multiplexing is not limited to this. Why? Because it's a combination between performance of the gene editing technology that we master, performance of the gene technology that we master for knock-ins and knockouts, and then finally the mastering of electroporation technology where you can series the things one after another. So again, I strongly recommend, if you're interested into this topic, to come and see the new devices that have been built. If you want to know about the gene engine, come tomorrow and you will see how we can multiplex series of knockouts with high yields and high volumes of T cells for not only clinical supplies, but also for commercial supplies in the future.
And I have a series of questions here that come to either electroporation or TALEN knockout efficiency. And maybe to start with the series of questions here, is there one aspect you're pointing to where we make the most improvements on that will further our gene editing efficiency, either in between electroporation or the way we edit a genome with TALEN? And also the question on multiplex editing. Is there a threshold where we would say this is too many steps in one go, so to speak, to kind of stay under a threshold of translocation? I know this is maybe a series of questions, but between Raleigh and Paris, feel free to take those questions.
Well, of course, it's essentially a regulatory aspect that you have here. You can multiplex as much as you want in the same step, and after this, of course, it can reduce the yields and can likely, it's not related to electroporation, like the gene editing technology or electroporation technology or anything like this. It's like if you go in the nucleus and you cleave series, let's say 5 loci at the same time, well, you will raise significantly the amount of translocation. Is that the problem for an allogenic product? I don't know. However, there is, of course, some red flags that are raised at the regulatory bodies when they start seeing the number of translocation that raises up. Therefore, and that's why also it's extremely important to be able to master not only gene editing technology, but also the electroporation itself. You can do series of small number of subsets of electroporation in order to avoid this. The problem that you have when you do this is that the cell continues to divide in the meantime, and so the number of cells to electroporate augment in the time and diversify, and also you need to sort this. And that's where we're strong in doing this, because we're able to do this in flows, and that will be explained tomorrow.
Thank you, Andre. Another question is, like, in the current context of the COVID pandemic and the amount of resources that are reserved for vaccine development, are you happy about the way you designed this manufacturing process from A to Z in-house?
I'm going to say, sorry, I like always taking all these questions, but like very general questions. When we started this idea of building our own manufacturing capacity internally, we thought about it from A to Z. It means from the buffer to the cell banks, to the plasmids, to the mRNA, to the vectors, and to the T cells. And that is revealed to be a fantastic opportunity. Imagine in 2020 or 2021 trying to order a plasmid or an mRNA, given the pandemic situation where mRNA is becoming a hot topic. We can do whatever we want internally. We're totally free. We don't depend upon anyone. And that makes a very key asset inside the company. We have huge capacity to produce all the mRNA, all the versions that we want, from the cell bank to the mRNA, to the cells, to the vectors. And yes, that has been a key event for the company. We haven't predicted this. But finally, we find ourselves in a very good situation today compared to all competitors, we believe, because everyone has a bit or piece of something, but here you have everything integrated from A to Z, totally completed and comprehensive inside the company. And I think that this was a great idea and a great asset in the future and a turn point for the company.
Thank you, Andre. I have a technical question from Wangzi Lee at Ladenburg. Thank you, Wangzi. And I would direct this to Steve in Raleigh. Could you talk about the adaptability of your manufacturing system and process, for example, to continuously increase automation or other improvement? Once validated, will it remain stable for years? And then the second question is, what's the advantage or rationale to separate the raw material and cell manufacturing facility into two locations versus nearby to make the transportation easier and faster? And that second question is a good one for Paris, for Leo. So maybe, Steve, you can start with the first part in terms of adapting the process and automation.
Sure. Thanks for the question. So the facility here in Raleigh was purpose-built for making allogeneic UCAR T cell therapy. So we do believe the process, when validated, is going to be very, very stable. The equipment qualification we've experienced so far has gone very well. The vendors that we've worked with, the partners in designing the equipment and the manufacturing train is very, very nicely designed, and we anticipate a lot of stability over the years and that sort of thing. The process is adaptable in the way that the process isolators we have designed are working, as they are easily reconfigured to other purposes by the nature of their design. So it's a matter of developing another equipment configuration. If something odd comes at us in the future, that's very easily adaptable to put it into the process isolator chain. So we have a lot of confidence in the stability of manufacturing and how robust the processes are going to be here.
Thank you, Steve. And Leo, what's the advantage to separate the raw material production from the final production and the two different locations between France and the US?
So thanks for the good question. So currently, that's our first approach. We have these two facilities, so we can concentrate our efforts on both in parallel. So currently, we have the facility for the starting materials here in Paris, and this is working very well. In the future, we will, also transfer those processes to Raleigh and have everything in Raleigh if needed. And we will use Paris for many other projects and also as a backup. So thanks for the good question.
Thank you so much, Leo. Maybe a few very specific questions here. What are the criteria to choose AT vials as primary packaging and not an IV bag? Maybe Steve or Leo.
Yeah, I'll pass that on to Tony Thatcher, our head of manufacturing sciences here.
Mm-hmm. Hi, yes, it's a great question. So the AT closed vial technology and the AT filling system is a very robust and scalable solution for products like ours. It was used during our clinical trials, and the real benefit is at small scale, our CMOs were using the manual filling system. We switched over to an automated filling system, which gives us much higher capacity, ability to fill many more vials, and also, as Steve mentioned, reconfigure for future products as well. But it uses the exact same filling technology and the exact same AT closed vials, so even though we have a much higher capacity, there's very little revalidation that needs to be done, and we'll get very consistent results with the data that we're gathering out of our earlier phase 1 clinical trial data, since we're filling and using the same container closure systems.
Thank you so much. Just one more quick technical question before we come to a wrap-up question. The technical question is how do you ensure the chain of identity during the manufacturing process?
Yeah, I mentioned that this is Tony Thatcher again. I mentioned that during the overview. It's less critical, obviously, for an allogeneic cell therapy than it is for an autologous. However, we do need to maintain our batch identity. As Dan mentioned, we need to be able to manufacture as many as 4 processes at once in a given isolator train. So we have a combination of electronic and paper-based systems that we use to manage the chain of identity through each batch.
and ensure that we have good segregation throughout production, and to give us that flexibility to do the number of processes that we anticipate being able to do here to ensure that we can manufacture all of our existing clinical trials and then expand and scale up for commercial production.
Thank you so much. And the final question we have time for today is from William Blair. Are your facilities large enough to support the commercialization of all UCART products currently in the clinic, as well as the new pipeline assets that we introduced yesterday? Steve, maybe you go ahead.
Sure, absolutely. That's one of the wonderful things about Selectus embarking upon internalizing manufacturing. The facilities were not designed as just clinical manufacturing. They were purpose designed for future commercial capacity. So yes, the short answer is we have capacity for the future commercial products.
Thank you so much, Steve. We're coming to the end of our time here, unfortunately, but we love the amount of questions we're getting, especially about manufacturing, which is an aspect that we want to talk much more about. So feel free to email me directly with your questions if we haven't answered them today, and we would be in touch with you directly. And again, thank you so much for all your time this year and this week to listen to our episodes. And please dial in tomorrow for episode 4, which will be another very exciting update on our capacity in-house and our technology.